Next, from Chicago, we offer an encore presentation of our behind-the-scenes tour of Chicago's Field Museum. This program, which was originally recorded in 2004, takes us to the upper floors of the Field Museum to see what research is conducted that benefits scientists from around the world. This runs about 1 hour.
So John, I appreciate you showing us behind the scenes of the Field Museum.
I'm delighted to do it.
We're on the fourth floor here. I guess this in effect is the attic, and certainly the part that the public never gets to see.
That's right.
This is where a lot of the researchers are housed and where they do their work?
Yeah, third floor, fourth floor, and what we refer to lovingly as the garden level, otherwise known as the basement, which is where my office is.
How many people are employed at the Field Museum?
About 620.
And all told, do you know offhand how much, it's a huge building, how much square footage you have?
It is 1 million square feet, but we are now adding another 180,000. So we're going to be almost 1,200,000 square feet on this location.
I know that so much of what the Field Museum does, people think of it as the Sioux, the dinosaur, and some of the mummies and all, but there's so much that goes on here, a lot of research that really adds to the knowledge and ongoing collections of what people know about any number of topics and fields of study. And that research is going on here. Really, it's kind of an international research center, isn't it?
Yeah, it's a major scientific research institution. There are 250 individuals here that are scientists on the scientific staff. 78 of those have PhDs. And we're involved in 94 countries around the world, with almost 23 million specimens now in the collection. So it is an enormous scientific research operation, and a very small portion of what we do really finds its way into the public view.
Well, let's begin our tour and we'll
Okay,
take a look.
great.
What are we going to see first?
Well, we're going to start up at the skins room, which is again just under the roof, so we are in the attic. And Larry Haney is a mammologist in the zoology department here. And Larry does a lot of his work in the Philippines, but he also works in the western United States. And he is one of the experts in the world of conservation biology. Not only is he able to study mammals and individual species, but the way that ecological systems all fit together. So one of his popular books that he's published is on the vanishing rainforests of the Philippine Islands. So one of the things that we do here is work very hard on our science, but then we also attempt to relate it to current critical issues of the environment. So Larry Haney is here. Dr. Haney, we are delighted to be here.
Thank you for taking your time on the Online Channel. What have you have a specimen out here. What can you tell us about it and how it matches with your research?
Sure. Well, actually today I'm helping out someone else with a research project. Not long ago we received a request for DNA material from grizzly bears in North America. This is a bear that is from just outside of Yellowstone National Park. It was shot by a group of hunters from Chicago who went out there 1902. This was the president of the Chicago Bar Association at the time. E.M. Ashcroft was the name of the person who shot this.
This is over 102 years old now.
It's 102 years old. This is an example of an animal that is from an area where the animals still exist today, but only in a very, very limited population. The reason why they're interested in getting DNA from these older specimens is that there's a fear now that with a small population of bears, there could be problems from inbreeding. And they want to know what the genetic structure of the population was 100 years ago. We have the specimens here in our collection that will allow them to answer that question.
So what research are you actually doing on this specimen at this time, or
well,
how is it aiding the...
Right. What I'm doing now is looking at the area here by the base of the paws to see if there are any scraps of muscle or other tissue that we can send to them so that they can use that material to look for DNA. The animal, the skin, like most of what I can show you here today, the skin has been tanned. And in the process of tanning, that makes it something that it will keep better for a longer period of time. It can be studied for a long time, but that tanning process destroys most of the DNA. But if we can find scraps of muscle around the toes, on the bones here, we can still get DNA from that 102 years later, even after the skin has been tanned.
What kind of information would you get from such DNA, and how would that add to your understanding of the species?
Well, as I mentioned, what we want to know about these grizzlies in Yellowstone is whether the small population, which now consists of certainly not more than 300 individuals in the entire population, is whether there has been loss of genetic variation. We know from lots of studies that when genetic variation is lost, often there's what's called inbreeding depression. Happens with dogs too. You can't continuously inbreed a population. When you lose genetic variation, you start getting more developmental abnormalities. Often the number of young produced declines dramatically. It's one of the biggest problems with maintaining small populations.
As I understand it, some of the species, in this case we have a grizzly bear, they used to roam on a much larger area in North America. Right. Now they're confined, I think you said, to 3
They're just
areas.
3 places in North America, in the United States, in the lower 48, where grizzly bears still have populations. The largest is the one that's in the Yellowstone area. There's another one up near Glacier National Park, and a third very tiny one in the Cascades in Washington State.
So as the development pushes these wild animals into more confined spaces, might that lead then to more inbreeding, which then by itself can also lead, if I'm understanding what you're saying, perhaps to the destruction of the population?
That's right. So even if the habitat remains there in the greater Yellowstone area, it may be the case that the number of individuals perhaps may be too small to remain healthy over the next 200 or 300 or 400 years, which is of course what we need to be thinking about. This is how we find out. We go back to look at the animals that were there 100 years ago and find out how much genetic variation was there then. We can compare that to the genetic variation that's in the current population and see if there's been a decline, whether we need to be concerned about that.
It's fascinating how these old collections, in this case a skin 102 years old, with the advent of new technologies, new scientific discovery methods, continues to add to our base of understanding of the species.
That's right. Well, that is one of the reasons why we want to be sure that these collections are here, not just 100 years from now, but 1,000 years from now. We want people to be able to come and use these specimens for research using techniques that we haven't even begun to dream about, I'm sure. 100 years ago, when this bear was shot, there was no such term as DNA. There was no awareness that it even existed. Certainly, the same thing will be happening in the future.
And as I understand, this is just a small part of your collection,
is that right? It's a tiny part. Would you like to see some more?
I would, yeah, sure.
Okay, let's go in here. Okay, come on in. It's a little chilly in here.
I was going to say, it is cool.
We keep this at about 50 degrees Fahrenheit and about 50% humidity all the time. Our biggest problem with this collection is, or at least our biggest concern, is having insects get in here. They could very quickly destroy the collection. And so, in fact, normally we don't let anyone in here except the researchers, people who must work with the specimens.
Do we actually, as myself, conceivably, are we bringing bugs into this room?
We absolutely could. And so we want to keep the numbers of people down as low as possible.
So even minuscule bugs can attack the collection.
Right. The ones that we worry about most are called carpet beetles. They're only, what would that be, about an 1/8 of an inch long, maybe a 1/16 of an inch, but they build up their populations very quickly, and they love leather. So they would destroy this collection very quickly if they were to get in here.
So by keeping it cool, it keeps them dormant.
Right, right. In the old days, we used to fumigate these rooms with cyanide gas to kill the insects. Oh. Obviously, we can't do that anymore. So,
how many? This is skin room number 1. It said on the door. How many skins are in this room?
Well, in this particular room, we have several hundred. The collection, well, actually, probably closer to 1,000. In the collection overall, we have probably about 10,000 of these tanned large mammal skins. So we have here.
And is this a lion?
This is an African lion. This is one that, well, let's see. This was taken by, in fact, the same person who shot that grizzly bear at Yellowstone was in Tanzania in 1929 and shot this lion. Most of the specimens that we have in here are from the period of about 1900 up to about 1950. This is at a time when these animals were a lot more common than they are today, and there wasn't the kind of concern about declining populations and extinction that there is now. There is, well, we wouldn't want to and we would not be allowed to make this kind of collection today. But here it is. It's of tremendous scientific value to the world community, and we want to keep this collection in perfect condition, or as close to it as we possibly can, for 1,000 years. So one of the reasons why we're willing to let people come in for a very short period of time right now is that we're getting ready to move them down to our new collection resource center that's down on the south side of the building underground. We'll have similar rooms in terms of climate control, but larger, so that we can let the collection continue to grow in a place that will keep them stable for another couple hundred years, we hope.
Great. Are any of the animal skins in this room, or have any of these since become extinct since they've been gathered?
Unfortunately, yes. In fact, probably many of these animals come from places where these animals don't occur anymore. The habitat's gone and the populations are entirely gone. In a few cases, it's not just the population, but the subspecies or entire species that's extinct. So, for example, this bear right here is a Mexican grizzly bear. That subspecies, that population that's designated formally well, it is designated as an extinct species. It's not an endangered species, but an extinct species. The last of the Mexican grizzlies disappeared in the 1970s. This particular one was taken in about 1910, when the animals were still relatively common. We have just a few specimens. A few other museums have them too. We can use those to learn a great deal about that extinct population. We've got lots of other things that are very seriously endangered animals. I can show you an example here if you'd like to see. It's a bit narrow back in here. Space is at a premium, always in a museum. This specimen is a giant panda. I can pull this out a bit so that you can see it better. This is the first specimen of a giant panda that came to the United States. This is an animal that was actually shot by Teddy Roosevelt's two sons. It was considered to be such an honor to be able to collect the first specimen of a previously unknown animal, unknown to science, that they arranged to pull their triggers at the same time so that they could share the honor. Now, obviously, attitudes have changed enormously in the 100 years since they shot this animal, or almost 100 years. This would never happen again, but it did happen. We want this animal to be here for a very, very long period of time so that we can use it and not have to go back out ever and get any more. Wonderful specimens that we have in here. This is one of the largest collections of this sort anywhere in the world. We have probably about 10,000 animals in these tan skin rooms. We have one of the probably the only 4 really large collections of this sort. It's very heavily used by researchers from around the world. We of course do use it for our own research programs here, but we also have people coming in from around the world on a regular basis.
Well, and that was a question that I had. And just to what extent do you cooperate with other museums and researchers around the world?
We consider this to be a global resource, and we do everything we possibly can to have people use it very actively. Of course, we need to safeguard the collections. We need to take good care of them, but to the extent possible, we want them to be very actively used. So we have a scholarship fund that helps to pay for visitors, from particularly students from within the U.S. and visitors from outside of the country, to come here to work with the collections. We send material out on loan very, very actively. We have something on the order of 200 scientific visitors coming in just to the division of mammals here at the museum every year to work with the collections. And that's in addition to our own group of about 15 very, very active researchers, curators, research associates, graduate students, postdocs, who are housed here, who are using the collection essentially every day as part of their research programs.
Well, Terry, the next stop is our mammal preparation laboratory. And here is where the final resting place of many of the specimens. So we receive a number of objects from the Brookfield Zoo and the Lincoln Park Zoo, as well as all around the United States, but also as well around the world. And on the world subject, Bill Stanley, who is the collections manager here in Mammals, is with us. And Bill had the good fortune to be raised in East Africa, in Tanzania, and has done a lot of work in Mount Kilimanjaro and the Eastern Arc Mountains. And Bill is just back from Africa, and I think has some specimens to share with us that he acquired on his latest trip.
That'd be great. Let's take a look.
So, Bill Stanley.
Bill, nice to meet you. Nice to meet you, Bill.
Nice to meet you.
What can you tell us about? Tell us a little bit about what you do, and how does it add to our understanding of the animal species that you work on?
Well, as collection manager in the Division of Mammals, I'm responsible for preparing, labeling, cataloging, installing mammal specimens from all over the world. As John mentioned, we get some specimens in the local zoo, like the skeleton of this gorilla, the skull of this bear, but the vast majority of the material that we are processing here are specimens that have been recently collected in a variety of areas all over the world: Madagascar, Peru, Philippines, and Tanzania. I just returned last week from Tanzania, where I conducted a survey of a mountain that had never been surveyed before in Mikumi National Park in central Tanzania, and collected several specimens of small rodents and bats. This is a rat that is endemic to Tanzania and southeastern Kenya, and had never been recorded from this mountain. And these are 3 different species of bats that were collected. We don't know exactly what species they are, and we won't know until we clean their skulls and skeletons. So these are the carcasses of the specimens that I collected, and this carcass is from the bigger bat that you see there. And we need to clean this carcass in order to determine what species it is.
Yeah, it's really hard to tell what that. I mean, it looks just like a collection, almost like mud in a sense. Is that flesh we're looking at?
It's flesh. Just like the T-bone steak you see at the store has a bone running through it and muscle around it.
This over here, of course, these are cleaned bones. How do you get the flesh off the bones?
We do it with the beetles that are in this room here. So if you want to take a walk, I can show you those right now. I'm going to put this in these beetles.
All right.
This is the domestic beetle room, and these are how we clean the skeletons of the vertebrate specimens. And so, as I open this tank, you'll notice that there are a variety of screen trays in here. In each of these trays is a specimen that has been placed in this morning. What you see on these carcasses are the adults and the larvae of a species of beetle called Dermestes vulpinus, common name carrion beetle. It's the larvae that do most of the feeding, and this rat will be a clean skeleton by tomorrow morning. And so here is something that was put in yesterday, and you can see that in fact it is almost ready to come out. The ribs are accessible, the vertebrae are visible, the caudal, the tail is all set, so the flesh has been eaten from the bones. These beetles won't eat living flesh. We actually have to dry the specimen slightly so that the beetles are attracted to them. So I'm going to put this bat carcass into this tray here, and tomorrow morning I'll be able to pull it out and examine the skull under a microscope and identify what species of bat it was I caught in Tanzania this season.
In fact, it looks like, what is that, a monkey skeleton over there?
This is a chimpanzee that was also from the local zoo. And when the animals die, they're brought here and processed and then cleaned so that they go into our primate collection, which is one of the largest and most diverse in the world. And scientists from all over the world will come to study that chimpanzee specimen or that bat skeleton in order to uncover the mysteries that still remain about the mammals that live on this planet. Whether the person's interested in functional morphology, whether they're interested in systematic relationships among different species, whether they're interested in ecology, people will use that chimp specimen or this bat specimen tomorrow, next year, and 500 years from now.
Does that come from just physically looking at the bones? Do you cut the bones in half, or do you do any kind of testing such as DNA that we were just talking about when we were just coming from the skin room or the hide room? What kind of techniques are used to gather information?
For the most part, examination, measurements, looking closely at the specimens. But we are also scanning digital images. Some people are taking little bits of skin for DNA analysis. Some people are measuring the teeth in order to get an idea of the diets that are involved. There's increasingly, as we develop these techniques, the specimens that were used in the 1800s are finding new uses with new scientific techniques.
Are you finding that the animal species, well, let me just ask an open question. What kind of things are you finding that perhaps were unknown 10, 20, 30, or 50 years ago?
Well, a lot of the new discoveries involve the relationships between and among different animal species. So, for instance, we now know that species A of this mouse is more closely related to species B than it is to species C, which is something that we didn't know, and it's very important to us in terms of conservation, in terms of systematics. But we also are learning a lot more about the ecology and the natural history of these different species as well, based on scientific investigations that are on now.
Do you find anything, do you ever come across that perhaps in some cases some of the animals are picking up pesticides in their bodies or different kind of chemicals? Is there any kind of a strain being put on the animal species from pollutants perhaps being carried in the winds?
We ourselves aren't doing that, and I should point out that in fact, as a collection manager and as a collection here, my main responsibility is housing and archiving this material. Think of me as a librarian, and so the animals that are brought here are essentially books, and I put them away. It's the reader, it's the scientist that come and conducts investigations like the one you're talking about.
That's a good analogy. I can understand that.
So I take care of the specimens, and I'm doing my own research on the biodiversity of small mammals in Tanzania. But I will pull out a specimen and hand it to the scientist who is conducting the pesticide study or the other study you're talking about. I'm happy with that. One of the things that we've noticed, for instance, is in domestication, animals that are domesticated have weird morphology on their bones. They'll have excess bony growths on their vertebrae. They'll have holes in the palate. So there's an interesting thing going on with domestication of animals that you don't see in the wild. And the reasons for that are still a little unknown. But these are the sorts of things that we see working around here on a daily basis.
Are we still finding animals that we didn't know were on the planet?
Absolutely. Every year we find new species. And what's interesting is we're finding new species not only by going out into the field and collecting them, going out to mountains and forests that no one's ever been to before, and collecting these new species, but even more interestingly, we are finding new species by just opening the drawers in the collection. So a shrew that was collected in the 1930s from India turns out to be the missing link between the shrews of the Old World and the shrews of the New World, and that was discovered just in the past few years. The specimen had been in our collection for decades, and so this is actually one of the most important aspects to this building, is it's housing a collection that is there available for scientists to come study now, to come study tomorrow, to come study 500 years from now. And it may be in 500 years, the specimen that's in the collection right now will be revealed as the most important specimen of a very significant study. But it's waiting for that scientist to come and study it.
fascinating that, as you say, we just keep collecting these collections with the idea. In fact, we were just told that in another area of the museum, how now with DNA development, we can do tests that on some of these specimens that were collected 100 years ago, and they had no idea at that time that DNA would ever be developed.
That's exactly right. But I think what's another important aspect of this discussion is we are collecting this material, and we cannot even begin to study every aspect of them that we're collecting. We are housing it and archiving it and making it accessible to the rest of the world through the web, through having scientists to come study here. But what we desperately need are more scientists to study this material. And this is why there's such a concern in this, among this community and the museum community and the scientific community at large, that, that we want kids and young adults to become more interested in the stuff that we're studying so that we can train these future scientists.
It has a particular aroma in this room that our viewers won't be able to experience. I guess you might say, what, the smell of rotting flesh, is it?
Yeah, but this is where we have our coffee and donuts every morning. Now, the same beetles that do the work for us, if they got into the collection, it would be a disaster. And so we freeze everything for 24 hours before it leaves the room. And that's why we have the two doors. If these beetles got out and got onto those skins that I showed you out there, they would devour those. And so we make sure that any little stowaways on the skeletons are frozen before they leave.
Do the same beetles, are they used uniformly, or are there different kinds of beetles?
The same beetles are used around the world. They're used by forensic scientists. They're used, if I was to go to a natural history museum in London and ask, where is your domestic colony, the collection managers and the curators there would know exactly what I was talking about. The same thing anywhere in the world. You can get these beetles locally, or you could call up Carolina Biological Supply and get your own if you want to start a colony in your bedroom.
Well, I think I'll pass on that. But Bill, Stanley, thank you for sharing your knowledge with us. I think John wants to show some other aspects of the museum.
Thanks for coming by.
Thank you. John, it's amazing how much space you have up here that the public never gets to see, and really in this area it almost looks more like the kind of thing you would see in an old university.
Oh yeah, very much so. This is 1921 is when we moved into this facility. The museum started in 1893 with the World's Fair, the World Columbian Exposition, as a way of retaining the collections that had been assembled for that World's Fair. And then we outgrew the building, so in the 1910s Daniel Burnham designed this building. His successor firm, Graham Anderson, Probst and White, oversaw the construction of it. We moved in 1921, and the corridor that we're here standing in is the Department of Geology. So we have a number of paleontologists who are on the staff here, working on dinosaurs, reptiles, fishes, amphibians, just across the whole range of fossil plants.
I started to say we're showing people behind the scenes of the Field Museum, but we're still only going to see a small portion of everything.
Right.
Otherwise, it would be a 10-hour program.
Absolutely.
What are just some of the different fields that you have up here, and even so that we can at least allude to them?
Sure. Well, the zoologists, you saw two individuals from the mammal department. But in addition, in zoology, we have insects, we have invertebrates, fishes, birds. So it's the whole range of the animal kingdom in zoology. In botany, we have not only flowering plants, we also have liverworts, we have fungi. So the whole plant kingdom, or fungi really is a separate kingdom at that end of the world. Anthropology, we have archaeologists who are digging in China, Peru, Mexico, Kenya, around the world, and a marvelous collection of pre-Columbian artifacts that we'll look at in a little while. And then here in geology, we study the meteorites, the hard rocks, and then also paleontology. So it's quite a university in a lot of ways. We do training of students, undergraduates, graduate students. We have wonderful relationships with the University of Illinois, Chicago. The University of Chicago, in our DNA laboratory, will have 27 PhD students from the University of Chicago whose degrees will be awarded there, but who are working here with our curators and with this collection. So it's a wonderful academic institution in addition to being a public museum. We're now in the Americas portion of that storage area, and As you can see, these collections are just so extraordinary and so extensive. It's mind-boggling. And when you think about the public part of the museum, our inability to share all of these wonderful objects with our visitors.
It's unbelievable just looking at the extent of this collection, how much there is. And as you said, I mean, I would have no clue when I would be visiting the museum in a normal sense that there would be this much behind the scenes.
Yeah. And this is very heavily studied by scientists and archaeologists, anthropologists from around the world who come to this collection to study it. So while the public doesn't get to see it, it is very heavily used really on a daily and weekly basis. And right here in the collection is Dr. Jenna Haws, who is the curator of American Ethnology and Archaeology of the Field Museum.
Dr. Haws?
Nice to meet you.
Nice to
Real
meet
pleasure.
You.
Welcome.
This is amazing, the collection you have here. Is this, no one would know it's this large.
Well, you can see this room goes on. There are 3 more rooms over this way, and this is 4 stories high. We have 3 of these collection storage areas with collections from all over the world. My particular area right now I'm working in is in Peru, and I'm looking at the beginnings of civilization in the New World. I'm looking at cultures that started around 3000 BC. Now, to give you an idea, this is before the first pyramids were being built. And we're looking at the beginnings of agriculture, we're looking at the beginnings of stratified society. And I use these collections to help me understand those.
And how do these lend to that understanding?
Well, I have a colleague with me right now who's working to help me translate a myth. And the myth was recorded by Spanish missionaries in 1638. And it came from one part of the Peruvian coast, one very small part. And in the myth, there's this wonderful story of first woman approaching the sun god and saying, "We're living on roots and spines. I need a son to help me rejuvenate the world." And the sun god gives her a child. But his brother, the evil Pachacamac, then is angered at the child, kills the child, tears him apart. His mother buries the parts of the child, and out of his teeth grows corn, and out of his bones grow potatoes and yucca, and out of his flesh grows fruits and vegetables. Well, we take that myth and we're able to look at the archaeological record from that same area and see that corn is growing there at 2500 BC, that fruits and vegetables are being grown. They're first being grown in this same location. So we have this wonderful connection between a mythology that was recorded in the 17th century and an archaeological history that goes all the way back to the 3rd millennium BC. And what I'm doing here in the collections right now is I'm actually looking for evidence of the manifestations of that first boy child who was born out of the pleadings of first woman to her sun god.
Because it being a deity, they would reflect that in their art.
In their iconography. And what happens is, we found in our excavations this last year, we found manifestations of a deity type figure. And here you see there's a figure here. He's got fangs. You see the fangs coming up here. He's got splayed legs. He's holding on to a post. Well, this is on a piece of gourd that we've dated to 2250 BC. That's an old gourd. And it's the first indication we have of a real supernatural being. You could say, well, you might ask, how do you know that's a deity? Well, clearly it's a supernatural figure because of its fangs and the way it's holding itself. Well, if you look at that, just its face, I can show you indications of that same figure that come later in time, come 1,000 or even 2,000 years later, where you see different manifestations of it. Want to see one of those?
Sure.
Let me show you. If you look at this figure, this is actually a figure from the Nazca culture. It dates to around 600 AD. So I'm going to let you hold it. Again, that's the behind the scenes. Now, if you look at its face here, it's got this headdress, it's got a nose piece coming down, the two eyes, but then look particularly, it's got fangs coming down here and a fang there, and then two fangs coming up. Well, here you have the headdress, the nose piece coming down, three fangs down, two fangs up. This is the same deity figure that we're seeing here, only you're seeing it being manifested over thousands of years in India and South America. So that's one of the things. I'll take this for you before we both drop it.
Thank you. What is the age? How does this piece of pottery correspond with the age in the book?
This is 2,000 years later. So what you're seeing is the same deity being followed out, being elaborated, becoming more sophisticated, and being manifested in different ways that we're now trying to understand. And we do it through excavation as well as through using these wonderful collections we have at the museum.
So as a layperson, when we're looking at this vast collection here of pottery, it's not just the pottery and as I would look at it from an ignorant base, if not understanding, but it's a reflection of the values, the religion, and various aspects. So am I correct that it would lend itself just to the whole understanding of the development of the society?
What you're seeing here is the history of peoples throughout the world manifested in our collections. You have certain written histories, but then you have unwritten histories, and we're trying to interpret those unwritten histories of many different parts of the world, and these collections prove to be an invaluable resource. It's like a library of the past.
What is the range of age of the development, or what is the age range of your collection, I should say?
Well, we have the remains of Magdalenian girl, which goes back to probably 19,000 years ago, coming all the way up to materials that I collected from the Hopi Reservation in 2001. We have the full range of mankind here. Most of our collection was done where we were able to amass these great collections that you see here by work that was being done in the late 19th century and early 20th century. So the bulk of this, particularly the prehistoric collection, came in then. Today, countries don't like to have this material out. Much of it we've gotten through trade with the countries. Everybody knows we have it here, and they're all quite comfortable with it. But today, this represents really a unique resource here in the United States.
How many, do you know how many pieces are in the collection overall?
In anthropology, we now have about 1.6 million pieces represented.
Amazing.
And representing cultures from Asia, from Africa, from Australia, from the Pacific, North America, South America, Central America, any place you point your finger, pretty much, we have collections from that area.
As you mentioned, you have here, I believe, from Peru.
Yes.
That is now you're working together on the research into the development of that mythology.
That's right.
How many researchers offhand would come to the Field Museum over the course of a year that you might work with to look at the collection?
Well, in anthropology, we have 200 to 300 researchers using our collection on a regular basis. Other departments have, you know, the botany department has more, and geology has more. Anthropology, I will generally work with 30 to 40 researchers a year coming in to work with particularly the American collections.
Aside from religion, what are just some of the other things that the artwork here tells you about these societies?
Well, why don't we take a look at some of it? You can actually see that here you have a frog, and here you have a dog. They're depicting everyday parts of their lives. This particular material is coming from the Moche area, which is on the coast of Peru. And they represent all different aspects of their lives being manifested in their pottery. In a way, they're telling, they have an unwritten kind of history where they're telling you stories through their pottery. And we're just beginning to interpret that now. But if you look at the bowls, you can tell what they're eating. If you look at the size of the bowls, you can tell how many people are eating out of it. So is it a small little bowl, or is it big bowls? You have big servings. So you know how many people were participating in any particular kind of event. We're able to look at this and get dates from it. So we can tell by the shape, for example, of a particular spout. From looking at this spout, I'm able to tell you that this particular vessel was made between 400 and 500 AD because the spout style changes over time. We're able to tell you where the clays came from. There are many aspects of life that we're able to extract out of these collections.
It's a fascinating story. I wish we could have more time with it.
It's a real pleasure talking about it. Obviously, I enjoy it. I'm enthusiastic about these collections. They're one of the real joys of being a curator here at the Field Museum.
Well, thank you for taking time to talk to us.
My pleasure.
It was great meeting you.
You bet.
Thank you.
Well, Terry, we're now in the fishes department. We're down in the basement of the Field Museum, or as we refer to it, we call it the garden level of the Field Museum. And our fishes collection here is managed by Phil Willink.
Hi.
So,
good to meet you.
Good to meet you.
So you have quite a collection here.
Yeah, this is a collection of fishes that we made several months ago in the country of Suriname, South America. It's a region where absolutely nobody lives, not a single person, not even indigenous people. It's totally uninhabited and one of the most pristine areas on the planet that we've ever seen. Some of the fish that we collected here, I'll show you some more in a few minutes. This one's called a Hopleus, or in the local language, Ayamara. They're 3-4 feet long, and they're a very popular food fish. They fish all the time. And the way they fish for them...
This looks like a snakehead. I mean, just
it's somewhat similar to that in body shape, yeah. But they use great big hooks with a hunk of meat, with a piece of rope, and they throw it in the river and catch it and eat it.
Those have some teeth on that.
Very big teeth, very big teeth.
Do they ever attack people?
They say they do, and they probably do if you're not careful. But if you know what you're doing, you'll be fine.
Is that something that can be turned around?
Oh yeah, sure.
How large would that fish be as far as, you say 3 to 4 feet?
They can get 3 to 4 feet that we know of, and they're very abundant. They're very abundant up there, one of the top predators.
And in what body of water?
It's called the Coelacanth River, which is a large river flowing into the Atlantic Ocean in northern South America.
This is, I gather, just a small part of the collection. You know how much the collection that you have?
This is all that's staying here, and another portion of it will be staying in a museum in South America, in the country of Suriname. We split them that way because it was a joint expedition between us and people there.
But in the overall in the fisheries department here, how large is the collection?
Oh, the collection's much larger. We have over 1 million fishes in the collection.
Is that right? And when you, over what period of time have these specimens been collected?
We have specimens dating back to the late 1800s.
Sample right here.
Yeah, this is called the Goliath Grouper. You can catch these off of Florida, the Caribbean, Gulf of Mexico. This fish in particular is probably about 5-6 feet long. He's very big. You'll find them swimming around coral reefs.
Of course, as you say, these are still available.
They're still there. Some other fish that I sort of pulled out right now. Here's called the Porcupine Fish. This is actually a model, a dried skin, like that. And normally they're deflated, but when they get scared, they'll suck in water and they puff out big. So it's actually twice its normal size right now, and covered with spines so that other fish don't eat it. One of those fish that could possibly eat it would be this right here. These are the jaws of a bull shark, and I think everybody knows that what they look like from this side. But what most people don't realize is if you look at them from the inside, it's just rows and rows and rows of teeth, hundreds of teeth. And what happens is when one of these on the outside chips or breaks off, it falls out and the one behind it snaps up into place. So they always have sharp teeth and they're always ready to bite or eat or do whatever they want. It's like a conveyor belt. This was collected in a lake here in Chicago in 1899. Okay, over 100 years. And basically, this is a unique fish. I mean, nobody else has this fish. I mean, they have representatives of these species, but not 100 years old. So what we can do is go through our collection and we can see all these old fish and what was caught when, and we can figure out what these areas were like 100 years ago, 150 years ago, and then we can look at them 80 years ago, 50 years ago, and we can see how these things have changed. And
how would having this fish and knowing what area of the lake it was caught in, how does that lead you to have a better understanding of what the environment or was then?
We can recreate the ecology of the lake. As a matter of fact, this was the site of a bio blitz in the year 2002 that was helped organized by the Field Museum. So we go there today and we know what the lake is like today. It's 18 feet deep with a sand bottom and stuff. It's Wolf Lake. But we know in the past by looking at the fish that the lake was actually only 3 or 4 feet deep and it was mostly mud and vegetation. And the fishes there were very different than the fishes that are there today. And because of all the homes that have been built up around it and factories and interstates and all that sort of stuff, the lake and the fishes have changed. And we can recreate that because we have fishes from 100 years ago here in the collections.
I'm waiting to see what you're going to show us here, Phil. This better be good.
This is a very unusual fish.
One you say was thought to be extinct and then was discovered it wasn't.
This was a fish known from fossils, and these fossils are about 400 million years old to about 70 million years. And then everybody thought they were completely extinct, no longer living on planet Earth.
How long ago did we think it had gone extinct?
70 million years ago was the last recorded fossil, until the year 1938, when a fishing trawler off South Africa caught one by accident. And at the time, nobody knew what it was. But after some scientific work, they figured it out, and what they decided is that it's actually called a coelacanth.
That's amazing.
These are fish. They live down...
There's actually two of them in here.
Yeah, we have two in here. They live down several hundred meters.
And we're getting quite an aroma coming off of this liquid. What is this liquid?
This is all ethanol. This is how we preserve fishes a long time, and from what we can tell, it'll keep the fishes like this for hundreds of years.
So is this one of the ones that were caught in 1938?
This is not one of those exact ones, no, but this is one collected in basically the same area. I can maybe lift it out a little bit so you can get a better look at what it looks like.
Pretty good sized fish.
Yeah, and what's unusual about it is even though we're calling it a fish, I want to point something out to you.
It almost has legs on it.
Those are legs. Now, I'm calling it a fish, and most fish look something like this, if you can see that. See how the fin splays out as a fan straight from a base?
Yeah.
This one right here has a short arm and then a fin. So these actually are arms. These are not really fish. They're actually in between fish and amphibians.
Amazing.